Just 6 elements make up roughly 99% of your body weight: oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. Understanding how these elements interact is the foundation of comprehending life itself, and it's what we explore today through the lens of Professor Vanessa's insights into the chemistry of living things. Let's begin by defining some fundamental terms. Chemistry is simply the study of matter and energy. Matter, as Professor Vanessa explains, is anything that has mass and occupies space. It's composed of elements. Energy, on the other hand, is the power to do work. Elements are the fundamental, pure forms of matter. You can't break them down into simpler forms. We find all known elements listed on the periodic table, which lists them in order of increasing atomic number. Now, let's zoom in on the smallest functional units of an element: atoms. An atom consists of a central core, called the nucleus, which contains protons and neutrons. Protons carry a positive charge and have mass, while neutrons have no charge but also have mass. Surrounding this nucleus are shells filled with electrons, which have a negative charge and, crucially, no discernible mass. Each element has a unique atomic symbol, like Na for sodium or O for oxygen. The atomic number tells us the number of protons, which is always consistent for a specific element. Atomic mass is roughly the sum of protons and neutrons. In an electrically neutral atom, the number of protons always equals the number of electrons. However, atoms of the same element can sometimes differ. These are called isotopes. Professor Vanessa notes that isotopes have the same number of protons but a different number of neutrons, which means they'll have a different atomic mass. Unstable isotopes are known as radioisotopes. These radioisotopes give off energy in the form of radiation and particles. They have practical applications, such as dating fossils using Argon-14, diagnostic imaging, cancer treatment, and even powering cardiac pacemakers. Most stable atoms have all their electron shells filled and all electrons paired. But there are exceptions: free radicals. These are atoms or molecules with one or more unpaired electrons, making them highly reactive. They can damage proteins and DNA, potentially accelerating the cellular aging process. When atoms combine, they form molecules. A molecule is a stable association between two or more atoms. These can be atoms of the same kind, like in an oxygen molecule, or different kinds, such as water or table salt, which is sodium chloride. Energy, as Professor Vanessa highlights, fuels life's activities. We differentiate between potential energy, which is stored energy, and kinetic energy, which is energy in motion, actively doing work. Potential energy can transform into kinetic energy. Electrons also possess potential energy. Each shell around an atom corresponds to a specific level of this potential energy. Shells farther from the nucleus hold electrons with more potential energy. If an electron absorbs energy, it moves to a farther shell. If it moves to a shell closer to the nucleus, it loses energy. Chemical bonds link atoms to form molecules. Atoms are most stable when their outermost electron shells are full, so they'll interact with other atoms to achieve this. Chemical bonds are the attractive forces holding these atoms together. Professor Vanessa outlines 3 main kinds: covalent, ionic, and hydrogen bonds. Covalent bonds form when atoms share electrons, creating very strong connections. For example, a hydrogen molecule shares 1 pair of electrons, forming a single covalent bond. An oxygen molecule shares 2 pairs, forming a double covalent bond. Water also involves covalent bonds. There are 2 types of covalent bonds. Nonpolar covalent bonds occur when electrons are shared equally. Polar covalent bonds, however, involve unequal sharing of electrons. In a water molecule, for instance, oxygen pulls on the shared electrons more strongly than hydrogen does, creating a slight negative charge near the oxygen and slight positive charges near the hydrogens. Next, let's consider ionic bonds, which occur between oppositely charged ions. An ion is an electrically charged atom or molecule. If an atom loses electrons, it forms a positively charged ion, like a sodium atom. If it gains electrons, it forms a negatively charged ion, like a chlorine atom. An ionic bond is simply the attractive force between these oppositely charged ions, as seen in table salt, sodium chloride. Finally, we have weak hydrogen bonds, which form between polar molecules. Polar molecules, like water, contain polar covalent bonds that result in uneven charge distribution, even though the molecule is overall electrically neutral. A hydrogen bond is a weak attraction between these oppositely charged regions of different polar molecules. It's these weak forces between water molecules that give water many of its unique properties. Living organisms are remarkable in that they contain only certain elements. While there are nearly 100 naturally occurring elements, about 99% of body weight consists of just 6: oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. Life profoundly depends on water, as Professor Vanessa emphasizes. Water has several key properties. It's an excellent solvent, meaning many substances dissolve in it. It's liquid at body temperature, can absorb and hold a significant amount of heat energy, and its evaporation uses up heat energy. Crucially, water participates in essential chemical reactions. As a biological solvent, water dissolves other substances, which we call solutes. Molecules that are attracted to water and interact easily with it are hydrophilic. Those that are nonpolar, uncharged, and do not interact with or dissolve in water are hydrophobic. Water's liquid state at body temperature allows it to serve as a vital transport function in the blood, which is 90% water. It's also the main constituent of both intracellular and extracellular spaces, making up about 60% of our body weight. Water also plays a significant role in regulating body temperature. It absorbs and holds a large amount of heat energy with only a modest temperature increase, which prevents rapid changes in body temperature. Moreover, evaporative cooling, like sweating, enables the body to lose excess heat quickly. In terms of chemical reactions, water is central. The synthesis of carbohydrates, proteins, and lipids produces water molecules, while the breakdown of these same molecules consumes water. Another crucial aspect of biochemistry is the importance of hydrogen ions. A hydrogen ion is essentially a single proton without an electron, and it's one of the most important ions in the body. An acid is any molecule that can donate hydrogen ions, increasing their concentration in a solution. A base is any molecule that can accept hydrogen ions, decreasing their concentration. Acids and bases effectively neutralize each other through their opposite effects on hydrogen ion concentration. The pH scale expresses this hydrogen ion concentration. It's used to measure the acidity or alkalinity of a solution, ranging from 0 to 14. A pH below 7 is acidic, 7 is neutral, and above 7 is basic. The higher the hydrogen ion concentration, the lower the pH, and conversely, the lower the hydrogen ion concentration, the higher the pH. To maintain stable pH in body fluids, our bodies rely on buffers, which are substances that minimize pH changes. Professor Vanessa notes that carbonic acid and bicarbonate form one of the body's most important buffer pairs, helping to counteract both excessive acidity and alkalinity. Moving to organic molecules, these are defined by Professor Vanessa as compounds made of carbon and hydrogen. They may also contain other elements like nitrogen, oxygen, phosphorus, or sulfur, and are held together by covalent bonds. Organisms contain 4 main types: carbohydrates, lipids, proteins, and nucleic acids. Carbon is the common building block for all organic molecules, making up 18% of the body by weight. Carbon's unique ability to form 4 covalent bonds allows it to create single or double bonds with hydrogen, nitrogen, oxygen, or other carbons. This versatility leads to linear, branched, or ring-shaped molecules capable of building both micromolecules and macromolecules. Macromolecules are synthesized and broken down within cells through two key processes. Dehydration synthesis removes the equivalent of a water molecule to link smaller molecular subunits, called monomers, together to build larger macromolecules, or polymers. This process requires energy. Hydrolysis, on the other hand, adds the equivalent of a water molecule to break apart macromolecules, releasing energy. So, dehydration synthesis is the reverse of hydrolysis. Carbohydrates are essential for energy and structural support. They typically follow the general formula with a backbone of carbons and hydrogen and oxygen attached in the same proportion as they appear in water. Their primary functions include serving as an energy source for most organisms and providing structural support, like cellulose in plant cell walls. Monosaccharides are simple sugars, such as glucose, which is a key energy source for cells, as well as fructose and galactose. Ribose and deoxyribose are also monosaccharides, found in RNA and DNA, respectively. These glucose monomers can join to form more complex carbohydrates. When two monosaccharides link together via dehydration synthesis, they form disaccharides, like sucrose, which is glucose plus fructose, maltose, which is two glucoses, and lactose, which is glucose plus galactose. Polysaccharides are thousands of monosaccharides joined in linear or branched chains. They function as energy storage, such as starch in plants and glycogen in animals, and also provide structural support, like indigestible cellulose in plant cell walls. Lipids are a crucial class of molecules that are insoluble in water, meaning they're hydrophobic. Professor Vanessa identifies three important classes: triglycerides, which are energy storage molecules, phospholipids, critical for cell membrane structure, and steroids, which are carbon-based ring structures. A key distinction of lipids is that they're the only class of macromolecules not assembled by joining monomers to form polymers. Triglycerides, also known as fats and oils, are composed of glycerol bonded to three fatty acids. Fatty acids can be saturated with all single bonds between carbons or unsaturated, including some double bonds. Triglycerides are stored in adipose tissue and serve as a vital form of energy storage. Phospholipids are the primary component of cell membranes. Their structure includes glycerol plus two fatty acids and a phosphate group. One end, containing the phosphate and glycerol, is water-soluble or hydrophilic. The other end, with its two fatty acid tails, is water-insoluble or hydrophobic. This unique structure allows them to form the bilayer of cell membranes. Steroids are characterized by their 4 carbon rings. Examples include cholesterol, vital for cell membranes and hormone synthesis, and hormones like estrogen and testosterone. Proteins are complex structures constructed of amino acids. They are long chains or polymers of monomer subunits called amino acids. There are 20 different types of amino acids, each with an amino end, a carboxyl end, and a unique R group. Amino acids join together through peptide bonds, which are formed by dehydration synthesis reactions. A peptide bond forms between the carboxyl end of one amino acid and the amino end of the next. A polypeptide is a polymer of 3 to 100 amino acids. A protein is a polypeptide longer than 100 amino acids that has a complex structure and function. Protein function is critically dependent on its structure, which Professor Vanessa explains at 4 levels. The primary structure is simply the amino acid sequence, stabilized by peptide bonds. Secondary structure involves the folding of this chain into alpha helices or beta pleated sheets, stabilized by hydrogen bonds. Tertiary structure refers to the protein's 3-dimensional shape, stabilized by a combination of covalent and ionic bonds, hydrophobic interactions, and hydrogen bonds, creating distinct polar and nonpolar areas within the molecule. Finally, quaternary structure occurs when two or more polypeptide chains join together. A crucial concept for proteins is denaturation, which is a permanent disruption of protein structure. This can be caused by high temperatures or changes in pH, and it leads to a loss of biological function. Enzymes, which are composed of protein, facilitate biochemical reactions. They act as biological catalysts, speeding up chemical reactions without being altered or consumed themselves. Without enzymes, many biochemical reactions wouldn't proceed quickly enough to sustain life. Maintaining homeostasis is essential to preserve the proper shape and biological activity of enzymes. Their functional shape depends on factors like temperature, pH, ion concentration, and the presence of inhibitors. Nucleic acids are responsible for storing genetic information. These are long chains, or polymers, made up of monomer subunits known as nucleotides. There are two types, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. DNA contains the instructions for producing RNA, which then contains the instructions for producing proteins. Proteins, in turn, direct most life processes. So the information flow goes from DNA to RNA to proteins. Nucleotides are the building blocks of nucleic acids. Each nucleotide contains a 5-carbon sugar, deoxyribose in DNA or ribose in RNA, a nitrogenous base, and a phosphate group. The structure of DNA is a double-stranded helix. Its nucleotides contain deoxyribose as the sugar and 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. These bases pair specifically across the 2 strands, adenine always with thymine and guanine always with cytosine. RNA, in contrast, is typically single-stranded. Its nucleotides contain ribose as the sugar and nitrogenous bases adenine, guanine, cytosine, and uracil. Notice that RNA uses uracil instead of thymine. Finally, we have ATP, or adenosine triphosphate, which carries energy. ATP is a nucleotide and serves as the universal energy source for cells. The bonds between its phosphate groups contain potential energy. Breaking these bonds releases energy, powering cellular activities. ATP can be replenished using energy released during other cellular chemical reactions to reattach a phosphate group to ADP, or adenosine diphosphate. This detailed exploration of the fundamental chemistry of living things from atoms to complex micromolecules was all based on the excellent lecture presentation by Professor Vanessa. If you found this discussion illuminating, please consider sharing the episode with someone who might also find it valuable.